Alright, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning or afternoon. Welcome back. Today we're gonna continue with our third lecture. Because it's a recording, so it will be a synchronous lecture. Now, if you look at the lecture notes, it's actually a bit long. There's 44 slides, and it will take a while to actually complete everything. So worst case scenario, we'll finish everything in one go. Otherwise, the lecture will be split into two lectures, lecture 3 and lecture 4. We'll be using the same lecture notes. And of course, if this goes into lecture number 4, I will add in new notes, okay, so that we will have enough time to actually cover everything. Okay, so before we start, we will do some quiz. Okay, so I'm hoping that you guys are actually viewing this from Spectrum. Okay, because as I mentioned, if you view it from Spectrum, there will be some questions, like a tutorial like, whereby I'll be asking questions and you will be able to practice and trying to figure out what is the correct answer. Okay, so last week we've looked at a few chemical ways to produce amino acid. Sorry, not last week, on Tuesday. We've looked at a few ways whereby you can manufacture or synthesize amino acid in the lab. Okay, so we are not going to review that one. I will just leave that as part of the questions. Okay, and we will just move into protein structure. Of course, if you're talking about protein structure, protein and peptides are different, in the sense that protein are more complex. Peptide is a subset of a protein, but that does not mean that the structures are totally different. So what we do know now is that protein is a bit more complex, and therefore there are 4 different levels of structural levels, as compared to peptides, which may or may not have 3 or 2 levels of secondary structures. Okay, so what we have known so far, what we have learned so far is multiple form of amino acids from protein. Okay, and of course, based on the general idea where you have a DNA, and then RNA, and then protein. Okay, as I mentioned from the first lecture, these are the original principle of biology. However, it's no longer true because some viruses utilizes RNA, for example, HIV virus, to produce a corresponding DNA, by which later it will produce RNA again and then protein. So the original RNA is a different type of RNA. Okay, so it goes back and resynthesize, not the virus itself. So the virus utilizes our body mechanism, our cells in the body, to actually produce DNA, and then reproduce more copies of RNAs, and then protein. Okay, so we have also learned that there are 20 naturally occurring amino acid that is being recognized by the IUPAC system. There are, of course, more than 20 types of amino acids if you consider the unnatural ones, and we've looked at the synthetic pathway by which you can synthesize amino acid. So basically, as one example is, as long as you have a carboxylic acid, you can pretty much produce any types of amino acids. Okay. Alright, so we also know that amino acid is the amino acids are the building block or building blocks of proteins. We will see why I say S over there. Okay, so by which you have one amino acid coupled to one another via the amide bond or peptide bond. Okay, forming the backbone of the protein. And of course, the R group are branching out from the I should have draw it like that. Okay, so the R group is branching out from the backbone of the protein. Okay, so that's the very basic general structure. So what we will cover next is the nomenclature of peptides, proteins, and amino acid. Okay, so we will learn them as we go a little bit. And the focus for today's lecture is to look at the different levels or structural levels of proteins. Okay, so generally these are the four levels. So first one, we have a primary structure, second one, secondary structure, tertiary structure, and quaternary structure. So those are the four levels. So basically primary structure, so quaternary structure built upon a tertiary structure built upon secondary, and secondary built upon the primary structure. Okay, so in general, primary structure is the sequence of the amino acid itself. Say for example, if you have multiple alanine, okay, so pentaalanine, so this is the primary structure, just the sequence of the amino acid. So when we talk about secondary structure, the 5 pentaalanine might form different structures such as alpha helix, beta sheet, coil or random coil, etc. Okay, so there are multiple known or identified some secondary structures in the protein, but what we'll be looking at is primarily just these three. So alpha helix, beta sheet, and coil. If you read journal articles, if you read the book, you might find there's a 310 helix. Okay, 310 helix. So there are a few types of other structure, but we will look at just these three. Again, in a little bit of detail, but not too detailed in the sense that you need to remember the sequence and so on and so forth. Okay, so from this secondary structure, you will build upon a more unified or a more complex three-dimensional structure. Okay, so assembly from two or more secondary structure, but the critical bit over here is that the tertiary structure is built upon similar peptide chain. Okay, meaning that these three are linked to one another. Okay, so from the sequence, it forms a secondary structure, say for example alpha helix, and then from this alpha helix, if you have an alpha helix and then a random coil and then a beta sheet, for example, then this is what we call as a tertiary structure. Okay, and then quaternary structure is when the 3D structure or the complete protein is formed by two or more polypeptide chain. So meaning if you have that as the first polypeptide and it is actually interacting with another protein chain, okay, to actually have a function. So this is what we call as a quaternary structure. Okay, so in basic, we will look at all of these in a little bit more detail. Alright, so primary structure so are linear. Okay, so primary structures or primary structure is basically a linear sequence of amino acid in a polypeptide chain linked via a covalent bond or covalent bonds, so to say, because it's not just one bond, it's multiple bonds. Okay, so you can go back to lecture 1 if you actually missed this information or you might be confused about amide bond, peptide bond, and so on and so forth. Okay, the shape of a protein depends on the primary structure, the core, since the side chain and the amino acid arrangement sequence will determine how the protein folds. Okay, so an example here is insulin. Okay, insulin is actually made up of two different polypeptide chain, so we call it chain A and chain B, and this is the sequence for chain A, whereby you have the C terminal over there and terminal over here. Okay, similarly for the chain B, and if you think about it, each one of these are covalently linked. Okay, so what I will do is I will use insulin as an example of protein. that has all four levels of structure. Okay, so insulin is not a very huge protein, but nonetheless, um, it is still a protein, and this is one of the example of a unique protein by which the number of amino acid is less than 100. So it's not very huge, but it still play a very critical role in our body. Okay, so that is the basic structure. So when you look at the basic structure, it's just the sequence. Okay, so when you look at the secondary structure, it refers to the spatial arrangement of that particular primary structure can take. So in a different words, conformational. Okay, conformational. It's a three-dimensional shape of that particular peptide sequence. Okay. The second structure can also be described describe how the segment of a protein backbone fold. So if you just look at the backbone itself, of course we will not know this unless if you do have the sequence. Okay, over there in green, actually the sequence in green forms alpha helix. Okay, while the rest of the sequence doesn't really form anything. Alright, so if you just think about that as a primary structure, it might seem like a linear structure, but then again, primary structure, we don't consider the three-dimensional shape of the amino acid itself. Once you go to a secondary structure, you can see that it's no longer linear and it's taking up more three-dimensional shape. Okay. So if you want to play around with this structure, what you can do is you can go to PDB, so protein data bank, okay, .org, and from here there are multiple things that you can do. You can just select a random protein looking at the structure, or you can also view the three-dimensional structure without having to download any additional app. Okay. All right. So as I mentioned in the secondary structure, there are three main substructures that we will look at. Of course, again, there's more than three, but the ones that we will be looking at is just these three. First one is alpha helix. Number two is a beta peptide sheet, or just beta sheet, and the third one is random coil, or some people call it just coil. Okay. So this structure of insulin shown over here is actually a quaternary structure. Okay, so the 4-degree. So you that on the one in green is one polypeptide, and the one in yellow is also another polypeptide. So when you have two or more polypeptides interacting with one another and having a certain function, then you can definitely say that they are quaternary structure. Okay, so without having this interaction, if you just consider that, then that one is just a the third level or tertiary structure of a protein. So you can view this information from this journal article. Alright, so from the figure of the previous slide, polypeptide chain are very flexible, but they are conformationally restricted. So what does it mean? It means that they adapt a certain configuration and tend to stay in that particular configuration. It might be a little bit dynamic, whereby it moves a little bit, but to the end of the day, it's a bit rigid, so it doesn't suddenly fold, becoming a linear without any three-dimensional structure simultaneously. Okay, unless if you boil it, if you put in chemicals, you break all the bonds, then of course it will change. We will look at that in more detail. So generally there are three factors that influences how peptides or protein fold. Okay, the first one is planarity region. Okay, about each peptide bond we will look at in a bit more detail. hydrogen bond, the strongest non-covalent bond in biology. So when we talk about the strongest non-covalent bond, then we will talk about hydrogen. Okay, but if you're talking about the strongest covalent bond or the strongest bond in biology, then of course it is the covalent bond. Okay, so a normal amide bond in this case, or even carbon-carbon bond. And the third one, the third factor that influences folding is steric hindrance. We will look at those three in a bit more detail. So what do I mean by regional planarity? So peptides or peptide bond or amide bond, this is also just a basic based on your first year chemistry, where when you have a amide bond like that, so amide has a lone pair, okay, as shown here, and therefore you can create a resonance structure. Okay, so something that has any chemical structure that has a resonance, the particular area are relatively planar. Okay, so similarly, if you think about benzene, okay, as compared to simply just a cyclohexane, right? So all of these has a pi bond, and therefore the double bond representation that we are normally drawn are rather inaccurate because there are resonance structures. Okay, so based on this resonance structure, it makes benzene flat, while cyclohexene, you can get the chair conformation, a boat conformation, and so on and so forth. Okay, so what I've mentioned here, a partial double bond character prevents free rotation about the peptide bond, planar, so it makes it planar. So the carbon and nitrogen and the other two atoms to which each is attached is highly rigid in plane. Okay, so if you imagine a piece of paper, right, and this is your amide bond, you can see that it's actually a bit thinner. Okay, I will probably show you guys some example in a little bit. Okay, can I show it to you guys? Let's see. So these are just some examples of the application that you can download if you have if you're using an iPhone or even on Android. You can actually use this. Okay, all right. Oops. Oh, I remember now. So this is should be able to see this model. Where are you? Unfortunately, I'm not able to show you guys. Never mind. All right. But if you go to again pdb.org, okay, supportingdatabank.org, find any structure, you can actually see that particular region over there is normally flat. Okay, but it doesn't mean that these other two bonds connecting to the amide bond are flat as well. They are relatively flat, but of course, because of the resonance, it's not 100% planar. All right, so a partial double bond, free rotation. Okay, so this is just another example. Again, example based on insulin. If we looked at this particular sequence over there, okay, cysteine, threonine, serine, and isoleucine. Okay, so that is the isoleucine. Oh, sorry, this is serine. Okay, isoleucine is over this side, so this one is the isoleucine, serine, threonine, and cysteine. Okay, and if you just look at the two amino acid, threonine and serine, so T and S in green. So if you rotate it towards this angle, okay, rotate it in about 90 degrees, what you can see is that the double bond over here and the amide bond are relatively planar. Okay, so imagine that all bonds are relatively like this. Therefore, the structure is a bit more rigid. It's not very rigid that it doesn't twist at all. It does twist, as you can see here. So it twists a little bit. Okay, the angle are off, but again, it doesn't allow for free rotation by which this can just turn any way it wants. Alright, so in contrast with peptide bonds, the bonds connecting to the alpha carbon are pure single bond. The two adjacent rigid peptide units may rotate about these bonds, taking on various orientation. This freedom of rotation about two bonds of each amino acid allow proteins to fold in many different ways. Okay, so what do I mean by that is this, if you consider the amide bond, okay, if you consider this amide bond in the middle here, okay, so this bond are freely rotatable. Okay, so this one and that one. Okay, so they can still rotate. Therefore, it allows a little bit degree of rotation, and therefore your peptide can actually twist or moves or whatever you want to call it a little bit. Okay, so freedom of rotation via the two adjacent rigid peptide units. Right, so the second factor, the first factor is the planary, regional planarity. Okay, so meaning it forms a planar, kind of like a piece of paper. Second one is due to the presence of hydrogen bonding, in maximizing the number of peptide groups that engage in hydrogen bonding. Therefore, it minimizes the overall energy. Okay, so the bonding strength of a hydrogen bond is between 7 to 40 kilojoule. Why does it vary so much? Well, it depends on the um, the distance between the two hydrogen bonds, the two hydrogens. Okay, no, not the two hydrogens per se, but the hydrogen and the um, atoms that can form hydrogen bond. So you have nitrogen, oxygen, sulfur. Okay, so that's the most basic one. So as long as you have um, these three atoms connecting to a hydrogen bond, so the strength of the bond can vary between 7 to 40 kilojoule. In contrast, covalent bond, if I'm not mistaken, is about 100, 140-ish kilojoule. It's probably more, kilojoule per mole, of course. Okay, so hydrogen bond, this is also a revision, forms between an hydrogen or a proton and electronegative atoms such as oxygen, nitrogen, and sulfur. Okay, so in terms of an amino acid, so you have all these amino acids that are polar or charged residues. Okay, polar, for example, serine. Serine only has an OH group, so we just consider that as a polar. But histidine, so these are all polar. But if you consider charged amino acid, for example, lysine, okay, by which you have an N terminal of NH2, so it can be protonated, so it can be charged. All right, so of course this one you can also consider it charge if you deprotonate the H, the protons over there, but unfortunately serine, in a biological environment, serine does not really deprotonate, so normally it will stay as an hydroxyl, and it interacts based on the polarity, but lysine, arginine, for example, or glutamic acid, they are normally the amino acid that plays a critical role in a catalytic activity of a protein. So therefore, even in nature, they can be protonated or deprotonated depending on the structure. Okay, and also you have the amide bond, and amide bond due to the presence of a proton on the end. Okay, so you can form a hydrogen bond. Okay, so either to a different amide bond adjacent to it in a different different sequence altogether, or it can be from the same sequence. Alright, so hydrogen bonding plays a very critical role in formation of all secondary structure, as you will see next. So in alpha helix, a coil in spiral is a subset of helix structure. As I mentioned, there's alpha helix, there's 310 helix, there's multiple types of other helix, but we'll just look at alpha helix, so to say. Okay, so L amino acid produces a clockwise coiled structure, or we call it as right hand turn, while D amino acid, as I mentioned last time, it's a mirror image, therefore it forms an anti-clockwise coil structure, if of course the sequence form alpha helix. If the sequence doesn't form an alpha helix, then it's not critical. But if the sequence form alpha helix, then this is the primary criteria whereby the L amino acid forms a clockwise rotation, as you see here, while the D amino acid forms an anti-clockwise, or the left hand turn. Okay, so what do I mean by anti-clockwise? If you look at from bottom like this, okay, so it goes that direction, while this one goes towards that direction. So one way to remember is using your hand and looking it from the bottom. Okay, so if you look at your hand and looking it from the top, then of course you will see that this one is anti-clockwise and this one is clockwise. Okay, but you need to look at from the bottom, and remember the sequence from the bottom to the top. Okay, like that. Right, so there are generally 3.6 amino acid residues per turn, and if you view it from the top, this is what you will see. All right, so this over here is approximately 3.6 amino acid, so forming one turn, meaning that from here until you form a circle, if you look it from the top, what you will see is you will see like that. Okay, so this is the same image as the previous one. If you take if you look at this sequence starting directly from the top, okay, top view over here, okay, one circle is actually from here going down into here. Okay, of course, if you're talking about right hand turn, then from the bottom going up into here. So this one turn rotation is about 3.6 amino acid. So a 310 helix has a different number of amino acid per turn. So alpha helix specifically has 3.6 amino acid. So additionally, if you're thinking about the hydrogen bond, because that is what we are talking about, okay, so you can see that each amide bond will interact with the amide group 3 residues above and 3 residues below. So if you consider that particular so this amide bond in the middle there, okay, so this amide bond interacts with about 3 amino acids below it. Okay, additionally, the carboxyl from the amide bond are interacting with 3 amino acids, the amide bond 3 amino acid above it. Okay, so 3 or 3 and above. So we just consider 3 because there's no 3.6 amino acid, there's 3 or 4. So this one is about 3 amino acid. Okay, so if you consider that one, 1, 2, 3, 4. Okay, so if you consider that, you will think 4 amino acid, but it's 3 amino acid above. Okay, so you don't consider the first one. So 1 amino acids, 2 and 3. Okay, so what else? So the hydrogen bonds, how does the hydrogen bond connects. So each peptide is trans and planar. What do I mean by planar? Over here is that alpha helix is circular, but the um, amide bond itself, as we've seen in the factor number 1, is relatively planar. Okay, the NH group of each peptide bond points roughly downwards, parallel to the axis of the helix, and the carboxyl group of each peptide bond points roughly upwards, also parallel to the axis of the helix. Okay, so if you look at this structure on the right hand side, it's actually inaccurate, so to say, because the carboxyl group is pointing downwards. So the sequence should be turned 180 degrees. Okay, 180 degrees. But this does not mean that the sequence is the amino acid or anti-clockwise, because towards the end of the day, to determine whether it's right hand turn or left hand turn, you need to look at the C terminal and the N terminal. Okay, so by knowing where the amino acid starts and where the amino acid is finished, you can actually go through and actually see whether the amino acid is turning clockwise or anti-clockwise. So over here it starts. The C terminal is somewhere on this side, and the N terminal is somewhere on this side. So that's why the structure shown here is different than what I've written here. Okay, so this, I'm just going to show you an example by which the figure might be rotated. So rotatable, so you can rotate the figure. But towards the end of the day, what's important is for you to understand the concept of the rotation based on where the protein starts and where the protein ends. The carbonyl group of each peptide bond is hydrogen bonded to the NH group of the peptide bond 4 amino acids units away. So it's more on three amide bond. But if you consider the amino acid, then it's actually the 4th amino acid. It's 3 and 4. So between 3 and 4, because amide bond is not from one amino acid, it's a combination of 2 amino acids. So yeah, so 3 amide bonds away, or the 4th amino acid. So all R groups points outwards from the helix. This reduces steric hindrance. So those are the R group. As you can see, they are pointing upwards. So none of them are actually pointing inwards. If it does pointing inwards, then it will break the structure. It will not be an alpha helix anymore. Right, so that leaves us to factor number 3, which is steric hindrance. Because of the R group pointing outwards, it gives out adequate separation between the neighboring amino acids. Neighboring, not I don't mean that it's adjacent to next to one another. This is I and I plus 4. Okay, so that's the principal concept, 4 amino acids away. All right, and by having this type of steric, it minimizes the repulsion of like charges. So if you have a lysine over here and a lysine over here, it can promote the formation of helix. If however you have different charges, so glutamic acid, for example, but a lysine over here, which will produce an attraction, and thus the helix might be bent a little bit. Okay, all right. So example shown here is leucine and leucine, leucine 3 and leucine 6, forming a steric hindrance in the insulin molecule. Alright, so how does it look like if you were to actually draw it in a 2D two-dimensional structure? When you consider the stereochemistry, then it will need to be drawn like this. So what you will see is that the amino acid forms a hydrogen bond. Wait, this seems like a beta sheet, but it's not. Okay, so supposedly it's going to the other one, but I can't remember why I put this structure in. But anyhow, I hope that this structure explains itself. And yep. Oh, okay. So this one is just a hypothetical. Okay, sorry, it's just a hypothetical. In cases whereby the R group are in the same side, okay, so like that. So you, of course, this structure has a very high steric energy. Okay, so that's why it says not formed because of steric hindrance. So this type of structure does not really form in nature at all. All right. We will look at similarly in the beta plated sheet. Okay, the steric hindrance occur, and I'll show you in the next slide in a bit more detail. So for a beta plated sheet, so there are generally two types of substructure, substructure, so to say. Okay, so this is a secondary structure, substructure of beta plated sheet, another substructure of either parallel or anti-parallel sequences. So what do I mean by that is that polypeptide chain running in the same direction. So if you consider that, say if the N terminal or C terminal is already shown over here, okay, so C terminal, N terminal over there, N terminal and C terminal over there, so therefore this one is anti-parallel and that one is parallel. Okay, so you can see N terminal, so C terminal to N terminal and C terminal to N terminal, so you can consider as parallel, while this one, oops, C terminal to N terminal and C terminal to N terminal, so it's anti-parallel. Okay, this is just a very simple basic concept. The CO group, the carboxyl group, carbonyl group from each peptide bond is hydrogen bonded to the NH group of peptide bond of the neighboring chain. Okay, so in contrast to alpha helices, whereby the hydrogen bonds are just 4 amino acids away, a beta plated sheet is for a neighboring chain. So this does not mean that it's from a two different peptide chain. Okay, it can be from the same chain, however they are further apart. Okay, as an example, if you have C to N like this, okay, that it goes around, going back, and then continue over here. Right, so they are still in the same chain, but they are in a neighboring. So this is what I call as neighboring chain. So it doesn't mean that it needs to be from a different peptide altogether, peptide chain altogether. It can be from the same peptide as well, but they are further apart as compared to hydrogen bond, alpha helices, whereby the hydrogen bond is just 4 amino acids away. All right, so this is just to show you in a bit more detail on the different structure of hydrogen bond formation between two different substructure of a beta sheet. So on the left hand side we have a parallel, on the right hand side we have an anti-parallel structure. Okay, so in summary of a secondary structure, in alpha helices you have the polypeptide backbone forming a spiral structure called alpha helix or helix simply, helix. The ones that we're focusing on is alpha helix. So alpha helix is term used for 3.6 amino acid residue per turn, or per circular turn if you're looking at the from the top. Hydrogen bond between stacking amide groups, again I to I plus 4, meaning 4 amino acids away or 3 amide bonds away. The R group projected outside of the spiral backbone to minimize energy. Okay, so if you have like that, so the R group are always pointing outside. In comparison, for a plated sheet or beta plated sheet, the arrangements of two peptide backbone forming sheet-like structure, two or one a very long one, of course. If it's just a short one, then this is very unlikely to happen. Okay, so you can form parallel or anti-parallel depending on the motion of where the C terminal starts and where the N terminal starts. Okay, so we need to also remember this. Hydrogen bond between the amide bond from one backbone to another. Okay, so again, even though it says backbone, it doesn't mean that it has to be two different polypeptides. As long as the backbone are further apart or further away from one another, then you can form a beta plated sheet. So the R group projected away from the center of the two sheet structure. So again, if you look at here, the R group are always pointing away. So this one is going up, okay, something like that. So it's always moving away from the center of the two plated sheet. The R group projected away from the center. Okay, the R group on any one chain alternates first above and then below the plane of the sheet, and so on and so forth. So what I mean by that is that if this one is bold, okay, so this one is dash. Okay, so it's an alternating. So towards the end of the day, they will have minimized steric hindrance. Random coil, however, I didn't show any because it's rather random. So why do I put in random? It's because random means that it doesn't form any distinguished structure. However, it's not so random in the sense that there are hydrogen bonds that keep the structure as it is. Okay, so it's not 100% free rotation. So there are still a limited degree of rotation. So it's random but not very random. Okay, so randomness is a definition only. As I mentioned, it's not really random. So some of the randomness serve as a joint for protein movement. So meaning that if you have an active site in the middle of the protein, however it is, so you will learn in SID2020 enzyme, so whereby I will touch about the flexibility of a protein. Then I will go back and relate to this lecture whereby the random coil structure of a protein actually allows a little bit of flexible, so the active site is not fixed. Okay, so it's a little bit flexible, it's a bit dynamic. Okay, so this is just to show you a different representation of the secondary structure.
Alright, that is all for today. We're gonna continue again next Tuesday. Okay, so I actually cut the lecture a bit short because we don't have enough time. Alright, thank you.